my name is Nicki Mosharaf, and I'm interning at the Botanigar Lab. So let's talk about what I worked on this summer. Many people may not realize how important trees are. Of course, everyone knows that they clean our air, but they also help cool our cities, and they do this through their shade and transpiration. The alarming problem, though, is that urban trees are dying at twice the rate of rural trees. This is because in urbanization, recent factors like heat have increased, pH has increased, human pollution has increased, and moisture has decreased, and that's causing a lot of stress to our urban trees. So to further understand how these factors are affecting our urban trees, we looked at the soil microbiomes, specifically the ectomycorrhizal fungi. Why are we looking at this specific sort of fungi? Well, that's because this fungi is actually really important in the soil microbiome. It basically helps facilitate a lot of interactions between the soil microbes, and it also helps transport nutrients from the soil to the tree. So it's definitely a key factor in the soil microbiome and also varies with urbanization. So we wanted to study how the soil was connected, how the soil microbes were connected in urban versus rural areas. And we hypothesized that if they're more connected, if the fungi are more connected in the soil, the tree basically has a healthier ecosystem and is a healthier tree. So that's why we hypothesized that in urban areas there's going to be less connectivity between the fungi than in rural areas. So to conduct our study, we gathered 8 soil samples from across different forests in Massachusetts, for urban, for rural, and we extracted the DNA. After taking that data, we used a program to create networks of the fungi and their connections. So how the networks worked is they calculated correlations between fungi. If fungi were increasing or decreasing in abundance, they were deemed as correlated. And if they were past a certain threshold of correlation, they were deemed as interacting. However, when we got our results, they were the exact opposite of what we expected. Urban areas were actually more connected between the fungi than rural areas. When we took a step back, we thought of why. Well, we realized that urban areas have tons of nitrogen, and this surplus of nitrogen is what the microbes feed off of. So in urban areas, the access to nitrogen allows many fungi to increase in abundance, and then the program sees, oh, these 2 microbes are increasing in abundance, and it deems them as interacting. So basically in urban areas, these microbes might be really highly correlated because they're all feeding off the nitrogen, but they're not actually interacting. So our next step is to add nitrogen in our graphs to see if these microbes are actually interacting with each other or if they're all connected to the nitrogen. So currently, I'm also extracting DNA from 2021 samples of not just urban forests, but actual street trees, the potted plants that we see all across our cities. And those data we're expecting to be even more extreme and our networks to be even more extreme because of the heightened urban conditions. So that's what we're headed for next.